At this time, I would like to turn today's program over to Canada Health Infoway. The floor is yours.
Thank you, Beatrice, and good morning, good afternoon to everyone. This is Fraser Rochford from Canada Health Infoway, and I welcome you all to today's webinar. As Beatrice said, this is a webinar about building a foundation for good analysis, and we're very pleased that you've been able to join us. You've dialed in. We also encourage you to tweet during this particular presentation. Our hashtag information is displayed there for you to use, so please feel free to do so. Also, just to give you a notice that this session is being recorded and will be posted on Infoway's website for later use. And as I will talk about a bit later, we encourage you to use, as Beatrice said, the Q&A feature to ask your questions, and we will do that throughout the presentation, so please don't hold your questions back, but we encourage you to ask them as we go through. In today's webinar, we'll basically be covering two topics and wind up with a further discussion. The first one will be a bit of background on the challenges themselves. The second one, we have a guest presenting the Building a Good Foundation for Good Analysis, and then we'll have a discussion. So, just moving right on into our overview of the challenges. As many of you know, Infoway partners with our different jurisdictions and other folks across the country to help accelerate the development, adoption, and effective use of digital health solutions. The Imagination Challenges, now in their, it's been 5 years since we started, seek to inspire, provoke, and promote innovation in health and healthcare and foster a community of innovators in Canada. We're actually really interested and particularly proud of the innovators that we've been able to foster when it comes to these data analysis challenges. So our first challenge, we had 40-some teams with 26 entries, and we're hoping to either better that in this Data 2 challenge. While each challenge is different, they all ultimately have the same goal, which is to improve the quality of care and the patient experience. We couldn't do these challenges without support from our supporting organizations. They're all listed here, and each one contributes in their own way, whether it be helping us to refine questions, as is the case in the Data Impact Challenge, helping promote the challenges through networks, and also acting as judges when it comes time to review the submissions presented by our participants. We've also had other organizations that have helped us refine questions, and together, all of these people help make these challenges possible. Now, the Data Impact Challenge 2, it's in full swing right now. It was launched on December 15th, and so many of you are probably have looked at the questions and are thinking about which ones you want to answer. And so this, we hope this webinar is timely for you to pick up a few pointers on how to sort of hone your analysis, and hopefully that can help you submit an entry into the challenge. There's over $95,000 awards available in this challenge. There's 7 questions for teams to answer. As you may recall, we started out with $85,000, and then we added an additional question, which bumped the total awards up to $95,000. Again, with anyone interested in the challenge, hopefully you'll find today's webinar valuable and help you create a high-quality entry. As we started off by saying, today we're going to have the lines muted, but we will be encouraging you to use the Q&A feature, and our presenter here is encouraging you to actually ask questions as we go through. So if you've got something you want to ask and it's relevant to what Jennifer's going to be speaking about at the time, please ask your questions, or if you have ones that you want to make sure they get answered by the end, submit them at any time and we'll make sure they get queued up. So without further ado, I'd like to introduce Jennifer Frood. She's a project lead with the Emerging Issues team at the Canadian Institute for Health Information. And prior to joining CIHI, Jennifer worked at Cancer Care Ontario and has been a professional researcher for over 15 years, covering a vast array of healthcare issues from cancer to chronic diseases to systems operations. She currently works with her Toronto-based team from her home in Vancouver. Welcome, Jennifer, and I pass this over to you, and I'm sure we'll hopefully generate a lot of fodder and thought as we go through your presentation.
Terrific. Thank you very much. Pardon me. So I'll just read it. My aim today is twofold. First of all, if you don't generally do research, my aim is to get you started on a good fitting. Or B, if you've been doing research for a while, provide a refresher on some of the basics. Even the best researchers can find themselves in the weeds if you try to speed through some of the foundational work. And having said that, I realize the deadline is coming up closer than I thought for Data Challenge 2. It is not too late to go back and check your foundation if you're well underway for your research already. Some of it is related to clarity of presentation as well as just making you think about some things prior to getting to the completed product. So analysis is a multi-step process with each action building on the previous action. So at the very top, who you drill down to or what your outcome will be depends on who you've included in the first place, and who you include will depend on your operational definition. I think most people can agree that the most exciting things in research are getting to that final answer, to get into the nitty-gritty of the findings, start pulling it apart in various ways. But realistically, about 80% of the work or effort needs to come in the upfront part, ensuring that you've got your final answer is built on a solid foundation, like good data quality, an outcome people care about. So you shouldn't get discouraged if you feel like you're spending a lot of time with your data before you ever get to the final answer. That's the more time you spend there and sitting with it and thinking it through, you're going to get a much better quality in the end, and you're going to have more options for drill down. You're going to find yourself less with what we call scope creep, which is one question leads to another question leads to another question. Before you know it, you don't remember why you started that in the first place. These building blocks help you keep on track. They also help you explain your answer better. Sorry, I just timed out here. My apologies. So if you can go on to the next slide, if you haven't already. So I mentioned a bit of this before. The first step in any good analysis is to provide or develop your operational definition. This is how you're going to proceed. This example I've stolen from Brandon, who's on paternity leave, but I'll thank him when he gets back. So the good operational definition will set the stage for your reader, guide decisions, highlight limitations. And the highlight limitations is important not to criticize your data, but to help you when you're comparing or understanding or interpreting your results. So how does one study differ from another? So if the limitation is that you weren't able to include in-hospital patient groups, that'll help you compare to other groups. So this example is one that we've taken from the Choosing Wisely work that Brandon's been working on. The question is, do not use atypical antipsychotics as a first-line intervention for insomnia in children and youth. Now, what I've underlined here are the parts where you will have to put an operational definition in. What do we mean by use? You know, atypical antipsychotics is a class of drugs. Do you want to use them all? Do you want to use one? What is first-line intervention? It's not mentioned here. And insomnia, chronic, acute, what exactly are we talking about? Now, some of this may be defined from your data, could be from the literature. It really depends on the data that you've got at hand and the data sources. And we can go on to the next one. So I've got a couple of hints here. Focus on the main question first. So when you're starting your analysis, again, it's exciting to get down to or to start pulling it apart and testing those hypotheses. Try putting blinders on. Focus first on the question you're being asked, and then you go from there. That'll help you with your scope. Parameters, sorry. The parameters, or how you operationally define it, will generally be defined by your data set. So in this example, this data comes from billing data or filling data. So when we say here, do not use, we only have dispensing data. We can tell what was dispensed. So that limitation or the parameter is defined by our data set. Sorry, so it's defined by our data set, so that's where that restriction is. The next one of atypical antipsychotic, this comes from exploring the data a little bit. Looked at frequency of which drugs were most common. And quetiapine was one up there, so again, so it's limited by the data set. And the other one is for insomnia. And in this case, I love this example because Brandon had to think a little creatively. In our data set, we don't have a diagnosis. We don't know why the drug was prescribed. But we do know that at low doses, it's subclinical, but it has a sedating effect. So using those two bits of information together, we can develop a proxy for the insomnia. I should also point out here that there's a line through the first line of intervention. Again, we don't have that information, so that parameter is defined by our data set. We won't be able to include that. I mean, the first line of interventions for insomnia are usually behavioral, which we don't get. So at the end, so we started off with do not use atypical antipsychotics as a first-line intervention for insomnia in youth and children. What we end up with, our operational definition of this, is do not dispense quetiapine at low doses for children and youth. So you can see how we took something, put our parameters on it from the data, and came up with how we're going to operationalize it. So we now know we're only focusing in terms of scope on quetiapine, so a single class of drugs, and in the low dosage level. So next, sorry, get my slides here. So define your cohort inclusion. So this would be the first, or one thing that you have to look at as well. Once you've decided how you're operationally defining it, you have to look at who are you including in that. Again, this will probably be dictated by your data. I will say when you write up your analysis, it helps to provide a rationale for why this group was chosen. There's no right or wrong answer, but it's good to clarify for your reader why you chose this. Was it the data you had limited for, or that was limited to? Is it something that's in literature? Is it a clinical question you were asked, that's the only group that they were interested in?
Jennifer, we do have a question here around, can you provide any guidance on how to determine an appropriate sample size when it comes to your cohort?
Sample size, as many as you can. So the more data you have, the more you will be able to do with it. So the larger numbers, you'll be able to start slicing and dicing into smaller cell counts. There is no absolute number, I'll say. There are. You can get into the power analysis. So do you have enough of a sample size where if you go and do a statistical analysis on it, that you will actually find something substantial? Those are sort of recommended. Those are usually done post hoc afterwards. So if you find a null finding or you found there was nothing there, is it because you weren't powered enough to see it? I would say target as many people as you can.
Is there any sort of guiding rule of thumb?
No. As a general number, no. Again, the numbers really become important when you start drilling down.
Okay.
So you can, if you're just looking for a rate of something and you have 200 people, then you can just do the rate. If you then want to start doing it by gender, you could start running into problems, particularly if you get small cell counts in one area. So say you've got 100 sample and you've got 20 women and 80 men. You're likely to run into problems there. So it's not a matter of an absolute number, but it's how they're distributed, what tests you're doing, what exactly you want to do with it, how far down do you want to take it. I would say the smaller number you have, the less drill down you can do. With healthcare data, though, it's interesting because most of the sample sizes, you have the opposite problem, where your numbers are too large. And I know this sounds odd. Coming from a university background, like when you're a student, you're working with samples of 30 undergraduates, right? What can happen in healthcare, particularly when you've got abstract level or record level data, is you are so overpowered that you're going to find differences where there may not be substantial or clinically relevant.
Okay.
Don't have the opposite problem.
Interesting.
Okay. So just going back to core, just a point here about definition and who you include. It's important to think about who you're going to be comparing to. So if you want to triangulate your results, it's better to pick a common definition where you can. And children and youth always causes us a little bit of problem, I have to say. Everybody can agree that a 5-year-old is a child. When it comes to things like 17, 18, 19-year-olds, are they in, are they out? It's good to think about who your audience is going to be. Are they pediatricians? What sort of group do they typically work with? And what sort of issues are likely to make a difference in where that age cutoff is? So I just encourage you to think about who is your audience, what are the common definitions in the literature. Some of this also may be restricted to the data, the way that your data is pulled in. Do you have each year individually, or do you have like 5-year age range? Do you only know the patient is between 15 to 19 years old? So there's limitations, but it's best to match as much as possible what's already out there. It'll just help with comparability and legs. If people can compare it to their study, they're more likely to take it up as well. Okay. Who will not be included? And I included this one because I think it's important to understand who is out as well as who's in. And again, this goes to comparability and understanding the generalizability of your data. Again, it's largely going to be restricted by the data that you have. So, for example, CIHI has in-hospital data. We have day clinics. We have ED, separated throughout Canada, better in some regions than others. We have very little on family practice. So if we're looking at a test, I know some of the lab ones, we might get data. Actually, sorry, the lab ones we don't get at all, but we may get some tests that are done in hospital, but we know that there's a huge chunk that are done outside of the hospital that we are not catching. And it's important to understand that bigger picture where your data will sit. You may also want to exclude patients with risk factors or other characteristics that make your patients distinctly different. So an example, at CIHI, we frequently exclude OB or general delivery from the general hospital patient because they're very different practice patterns, risk factors. And again, it will depend on the data and the question that you're looking at, but it's something to think about. And again, for your audience, who are they interested in? Sometimes it's better to have a homogeneous sample or to restrict it to a certain, like only cardiac patients, like to put those parameters around. You're going to get a cleaner, you're just removing white noise from from your sample, and you're going to get a cleaner answer. Okay, oh, and the poll. So again, I see that there's quite a few people checked in, attendees, and I'm just sort of wondering where people are coming from. So I'd like you to take the poll and just say, if you do you work with data sources where there is good documentation on data quality? Absolutely. No, the documentation's not good. I've heard of it, but I've never seen it. This is usually the stack of paper holding the door open. Or I don't work with data sources.
So our events people are just getting that poll set up, so soon you'll see that on the right side of your screen. There it is. If you'd please respond by clicking one of those boxes, that would be great. And then we can hit the submit button once you've selected it, and we'll be closing that poll in probably another 15 seconds. So quickly answer, please.
You need the Jeopardy music here.
You can hum it for us, Jennifer. Okay, let's close the poll. And it takes a few seconds for this to come up. Jennifer, any guesses? Where do you think people are going to stand on this?
If you were to take this strictly at CIHI, I would say that it's probably C. We have almost too much information, and it's kind of difficult to find things.
Ah, okay. All right.
The documentation is difficult in any organization, particularly when you have a lot of data. There's just so many ways to look at it you know, even if you have a lot of, well, I'll get into it, but I mean, even if you have a lot of missing data, it could be either unknown or it's documented, but it's still in there.
Right.
Left up to the researcher to figure out how they want to solve that problem.
All right. Okay. And here's the result.
I'm surprised that there's 46 in documentation. It's good. That's great. I think it's getting better with the electronics. I mean, when I first started, it was that stack of papers holding the door open. It was sort of there, but it was a lot to look through. With Google searches and things like that, it makes it a lot easier.
Right. So that means that like almost half of these people are going to have great data submit in the Data Impact 2 Challenge. So I think that's wonderful.
Perfect. Excellent. Okay, so that said, even if you have good documentation, we do our own pre-analysis data quality checks. Again, sometimes you could have documentation that a variable has a large amount of missing data, and it's just documented that it's there. You then need to determine what to do about it. And in order to determine what to do about it, you have to be aware that it's there. There are a couple of ways that data quality can get in or can impact you. Very often people say, oh, well, it's a mandatory data field. The data will be there. Just because it's there doesn't mean it's of good quality. So there are very simple, easy checks to find out what these are. One of them is obviously just a frequency check. Do the variables, are they within range? What tends to happen with mandatory fields is people need to enter something in order to move past that data entry point. So the one we always see is the year 999, which and if you anytime you see that, it's frequently what they've filled in for a default missing value. And this can cause problems because if you just pull data into your data analysis, like for example SAS, it'll convert that to a year without ever giving you an error or any indication that it's wrong. So unless you look at it or check it ahead of time, you will never know. So in some ways, the mandatory entry can cause as many problems as optional. So optional, it could just be missing. Mandatory, they're going to fill in something, not necessarily the right thing, but something, just to get past that field.
Jennifer, are there any tools to help make this easier to check that your data is of good quality or to improve the quality of your data?
So specific tools, no. The one thing, or the two things that I rely on most is A, your proc freq or your frequency count, and secondly is your eyes. So you'd be amazed when you look at your data, if you just sort of scan through, you will pick up patterns. So say you want to say you see that there's a lot of 999 and you want to go, what's going on? If you pull up that data, 999, and you can see just visually that they're all coming from one institution or one range or one sort of test, that'll give you a step of where to go. So proc freq was just simple. Take a look, high and low end, low end. Are they all a valid value according to your data quality documentation? It'll tell you how many you have missing as well. It'll tell you anything that's out of scope. I have a great example that we use of an error, not an error, but a challenge that we found at High, where we had high volumes of people being discharged at 23:59. So on a 24-hour clock, that's just before midnight. A valid time. You know, it's there, it's a valid time, but 90% of the people in their ED would be discharged at 1 minute before midnight. So that sort of gives you an indication that's not a good, there's something going on there. But unless you did a frequency counter or a check, you would never find that.
So Jennifer, does that mean you should discount that data, or what do you do with this once you've identified it as a potential problem?
Well, there's a couple of options, and it really depends on what you want to do with it. So if the missing data itself is not necessarily a problem. If you have like 10% missing data, it's completely fine. Like it's not going to cause any problems with your analysis. The challenge comes. Sorry, I locked myself out again. Remember to wiggle the mouse here. The challenge comes when the data is missing differentially or creates what's called a systematic bias in the data. So, for example, the 2359 that was all missing from one facility or two facilities, and they had no valid dates. Obviously, you can't include those. If you have a sort of smattering throughout, low volumes, you might want to just discount them. If you've got a moderate amount missing or invalid, you can impute if you have the right, or you have some good ideas of what that should be. Like you can impute it from the average time, you can impute it from the average of patients just like that patient at the same time. And that'll boost your numbers. There's no one quick solid answer. It really does depend on the context that you're doing it in. There's lots of literature out there on how to impute data. The only thing I would recommend or guidance that I would give is to make sure that it's not missing or invalid in a systematic way, because you're going to run into trouble there. Then you're essentially creating data for a certain group.
Okay, thank you.
Sorry, and just back on the pre-quality, I just want to point out that just because you have missing data or you have some invalid ranges you have to impute, don't throw the baby out with the bathwater. That your data is still probably really usable and really good. I would mention it though, because then you can somewhat let the reader decide. And again, this goes back to comparability. Let the reader make their own decisions. So don't let that throw you. If you think all the data is no good because it's got all this missing data, maybe you need to change your scope. Like you need to include only the other thing we'll do at CIHI is just include only larger facilities because some of the smaller ones have some data quality issues. There are ways around it. Changing scope is an easy one if you've got a systematic bias. Imputing is another one depending on if you're able to, I guess, is the low bottom answer. So don't let that throw you. Okay? So should have pointed out, pre-analysis is the one that you do before you start your analysis. There's data quality throughout, but the pre are getting the variables ready to be inputted and to be massaged and worked with. Post-analysis is a separate set of data quality challenges, steps to go through. First one, again, I'm a practical person. Face validity is huge. Do the findings make logical sense? Given what you know about the field, here's a good chance, or what you should do is talk to people in the field to see, do they make sense? Is this what you see? Particularly people who are on the field or on the ground level. Is this what you see in your day-to-day clinical practice? The other thing is, do differences between groups make sense? So I wouldn't expect to see, like obviously you're going to see, you shouldn't have any men in your obstetrics population. You probably see more men than women in your if you're doing cardiovascular concerns and things like, or more women in stroke. Like there are certain no gender differences. So just look at them and say, does this make sense with what I know of the environment? The second one is to triangulate outcome where possible. So here's where all that work on the operational definition is going to pay off. Does what you're seeing make sense with literature? And it could be another country, could be within Canada. Most likely it's going to be in another country, given the competition that we're talking about, and your research has not been done here very much. So does it match with any literature you can find? You can even triangulate within your own data set. Do you see wide differences between regions or population groups, depending on what you have? If you have two clinics that have very different quality, or very different results, does it make sense given what you know of the clinics? Talk to people at this point is what my advice would be.
Jennifer, my version of triangulation is I never trust one weather source, so I always have to triangulate with Environment Canada, the Weather Network, and one more source.
Yep. Always. So it's the rule of three, right? You can't just compare it to one either, because you're going to get two ranges, and then if you get the third one in between, you're on pretty sound quality there.
Oh, that's good. Now, Jennifer, also, so you've talked about it, it's important to do both pre- and post-analysis in your data quality. Is there one that requires more energy than the other, or the same amount of time kind of goes into both?
This, my gut instinct is to say pre, but that's really going to depend on how good your data quality is as well. So if you've got good documentation, you might spend less on the pre. If you've got a large data set or it's never really had analysis done on it, you're probably going to spend a lot more time there. And it also depends, again, on how much literature is out in the field. You could find that when you're doing a literature review, they all sort of come up to the same number. Very easy to check. Some of the challenging parts of the post-analysis, if the literature is all over the board, to compare to your number, you're going to have to really dig down and see what's most comparable to yours. You're going to have to read a lot of literature to try and make sense of it. So it kind of depends how much work has been done and how much documentation is on your end.
Okay, thank you. Just a reminder to everyone, you've been submitting questions. Please continue to do so. We've got Jennifer here who's a wealth of knowledge in this, and we'd like you to be able to ask those questions that will help you in your submissions, or whether just in your daily job, or whether it be you're thinking of doing particular analysis on something that's unrelated to our challenge. But Jennifer's here to provide you help, so please don't hesitate to ask questions using the Q&A feature.
Okay, thank you. This poll is sort of segueing into a bit of a shift in direction in terms of what you then do with the analysis. So again, because we're probably come from a variety of backgrounds, the question is really how often do you conduct data analysis for stakeholders? Is this something you do regularly, something you do only occasionally, you've never done it, or you don't know what a stakeholder is?
Okay, so let's open that poll, and everyone remember to pick your answer, A, B, C, or D, hit the submit button, and then we'll be calculating your results. So we've probably given them enough time now, Jennifer, so let's close the poll, please. All right, and we will wait for the answer. Any guesses on this one, Jennifer?
I'm going to go with occasional, although I would see a spike in don't understand as well. It's just, sorry, we're doing interviewing right now, and this is one of the questions we ask when we interview, and a large number of people don't really regularly talk to stakeholders, not even something on their radar.
Ah, okay. So, well, hey.
All the high people, isn't it?
So 63% of our respondents said that they are regular reporting. So how often do they conduct analysis for their stakeholders? Regularly. Interesting.
Excellent. So a lot of you are preparing material that will be taken up by other people, usually busy people. So this is my sort of goal with the next section is when you're presenting drill down and scope, to think about who your audience is going to be and to present it in a clear, concise way. People are busy, I'm not going to say lazy, but people are really busy. They take in massive amounts of information every day. The easier it is for you to be understood and to get what they need, the more accepting they will be of what you're trying to, the information you're trying to provide to them. Okay. So once you've answered your basic question, you can start then pulling it apart and investigating if you want to, which most people do, because again, that's where the fun stuff is. And here's where knowing your audience is useful to helping guide the direction of what questions should you answer, what will be of importance to the stakeholder. Like, we all have our own questions that we'd like answered. Take a minute to think about whether those same questions will be the ones your stakeholders are interested in. So there's a couple of ways that you can divide the data out. One is by risk factor. So who is at risk? Are you looking at patient characteristics? Who's at more risk for cardiovascular disease? Who's going to have a stroke? Who's having repeat lab tests? Is there something about those patient characteristics? The other way to look at it is from a physician or system characteristics. So are there policies or practice patterns that are in place that are more likely to result in repeat lab tests or more likely to result in a repeat visit to the ED? So there's two ways to sort of cut most healthcare data. The other thing to look at is for system characteristics by region, if you've got that option. Do regions differ? Is there something different about the practice there? Or it could be the patient characteristics within those regions that will make health regions look differently on paper. And again, some of this is going to be restricted to, your level of detail will be restricted by volumes and data, and that's where the number comes from. Small cell counts are problematic for comparison stability. So in some of our smaller provinces, if you've only got 10 patients that have this sort of incident you're looking for, or a test that you're looking at in the next year the doctor takes 2 months off on a sabbatical, their numbers will drop significantly. So there's a stability issue over time for some of the small volumes. And privacy problems, obviously. You're not going to report small cell counts or anything that where a patient can be tracked back so that somebody could identify that patient. So those are where your restrictions are in terms of volumes and where you can go with them.
Jenna, if we go back to the Know Your Audience, in the case of the Data Impact Challenge 2, the audience not only are the general public, because we do actually post all of the submissions that are qualifying onto our challenge site, but also ultimately judges will actually review these. And I think as a former judge, do you have any suggestions on like how do these people know, like, do you have to specifically know the panel of judges, or is it something that people are looking for in general when they're reading these submissions? So you did sort of mention the time and, people have to go through these quickly. As a judge, what were you really looking for when you went through this?
For the data challenge, I wasn't honestly looking for anything. The question was fairly vague. I was more looking for, did they know the data? What are the parameters of the data? Because that's really what the challenge was the source. It was a showcase of their source of data and what they could do with it. I was almost less interested in the results, but that's the nature of the data challenge. For the general population who are interested in the results, they probably have the same questions you do. There's usually some high-level ones that come up. So for the competition, not so much. At CIHI, because we have sort of strict stakeholders that we work with regularly, we know the questions they're going to ask. They're going to ask about peer group comparisons. They're going to ask about regional comparisons because that's what their job is. They're going to take that data and then report it back. For this one, the advice or the direction would be just to showcase the data. What can you do with it? Like sort of what are the parameters? That's somewhat up to you guys as well, though. Would you say you'd agree with that?
Well, certainly we set out rules that are that I encourage all participants to read on how the judges will be scoring the entries, and I think that hopefully goes a long way to say what, as an audience, they will be looking for.
Yeah, and sometimes you don't know your audience, even with us. If you're doing your report not to a regular stakeholder, there's a bit of, it's somewhat more freedom. You can sort of answer the questions that you had off the top of your head. Yeah, it's when you come with your regular stakeholders, they're the ones, like if you're targeting a very specific group, it's a little more restrictive.
Right.
If you've got docs or if you've got hospital administrators or policymakers, they may have some very different questions than a clinical group. So we're working with Choosing Wisely right now. It's taxing us a little bit in terms of it's a very different risk or stakeholder group that we're looking at. They want the clinical information. They're less concerned about peer group comparisons.
Now, when it comes to level of detail and restricted by volumes and data, we do have a question about what are some data privacy considerations when doing analysis that will be published? What do they?
Sorry, so how small can you go?
Well, or are there certain considerations that you have to do when you know that the data that you have will be publicized?
Yeah, so again, like I said, you want to make sure that any results will not be able to be tracked back to the patient. So at CIHI, for privacy, we've got a line in the sand that says no cell size can be smaller than 5. Now, if you've got So if somewhere you report that there are 6 patients in Canada that have this test and you're reporting all the provinces, it depends on the other information that you're providing in your write-up. You sort of have to be an investigator and say, try to track the person back. Can I find this person, or would the general population be able to find this person? If you've got like 1 or 2 people and it was a high-profile car accident, obviously that's going to be more of a privacy concern. So it really depends on how specific the data is. Like, are you talking about a very specific test? Are you talking about a test where you've got, it's hard to know without the actual data, but you've got something like, in PEI, you've got a 40-year-old male on renal dialysis. If anybody, it's small enough that somebody who's curious enough can probably find that person.
Okay, I think that will help the person who asked that question. If not, they will ask me a qualifying question. So,
yeah, if that doesn't help you, it's really hard. It is, like, again, we have a whole privacy team that looks at this because privacy is an important issue. And the challenge is it depends on what other information you're providing in the report.
Right.
And how much can people slice and dice it. Okay. Okay, so I've provided some drill down examples here, and I did take these from the Data Quality 2, the Data Challenge 2. So for an example of your adverse drug events, some things you might want to think about. Are there specific class of drugs or a specific drug itself that you want to focus on? Can you drill down, are there differences between drugs or classes? Are there regional differences? And that may go to SES or prescribing patterns. So commonly you see a north or a south difference as well as an east and west difference. And can you link that to SES? That would be a drill down question. Age of patient, comorbidities, all of those things are ways that you can slice and dice the data. And again, no right or wrong answers to these. It really depends on the audience or where or whether there's a story there that you can tell, or you have the volume, as we just talked about. So they're just things to think about. So same thing with youth and mental health. I thought this was sort of an interesting one. This one you can start drilling on the type of content because this is related to social media. You can look at the content of what they're posting, frequency of posting, profile of regular posters, and then your basic gender age differences. I mean, people generally go to gender and age differences, and they're useful to include, again, for that comparability piece. But the story is often in something else. It depends, again, very content specific, but don't think about it just gender and age, think about it in patient profile. And it may mean putting different variables together. So do you have older women? For your OB, is it a certain area where they're older? Is it a certain area, region where you have lots of younger moms and that's what's causing it? So it may not be just one thing, but combining elements as well. And I should also mention it's worthwhile showing null findings or areas where there's no difference. Again, as I've mentioned, high stakeholders frequently want to know about peer group. Even though there's no difference, we usually mention it because they're going to ask. So some of these things you can head off. Deal with the ones that people want to know, whether there's something there or not. Okay, the next one, this is really about presentation and ease of readability for your reader. Love graphs and charts. Most people are very visual, and they're a great way to transmit a large amount of information in a short period of time. So graphs generally work best. It's saying that not everything needs to be in a graph or a chart. Graphs do work best when they show relationships. They are great when you have a clear point. So you want to make sure that even the data that you're including in the graph has a story. You don't want to just throw everything in. Simple things like having check or have labels. Missing or bad labels can cause more confusion than clarity. Here's a good way to give this to somebody who's not naive to the project. Have them look at it and see if they get the 1 or 2 summary points. You'll see a lot of times in journal articles what they've started to do is you need to include a sentence or two saying, what is the chart about? It's a good exercise, even if you're not submitting it to a journal. If you can write it out in a sentence, your goal. that tells you that you've got it down to the bare basics of what you want to include. The next slide is an example, pardon me, of one where there is just too much going on. You can see there's a label missing here. It's got both genders, 30 days, 60 days. Like, there's just a lot going on in here. The general rule is if you can't understand a graph in 5 seconds or less, then it's not meeting its need. It's no longer a quick transmission of information. It's just taking the reader too long. As well, include the graph, but also include the synthesis in text. So you may present the information and the reader might interpret it very differently than you did. So it's useful, so don't think that the graph can't replace the text. It needs to be in conjunction with. And you know, after you do all this work, you take the time to spell it out and make sure that your opinion is being heard or your read of it is being heard.
So I think that 5-second rule is very, I'd never heard that before, so that's interesting. That's good. That's a good piece of advice for everyone.
There's a few of those around where being able to concisely communicate really shows people that you understand it. Again, they're busy. If they don't get taken in quick, they're likely going to move on to the next thing. Some people talk about the elevator rule, where if you can describe your study on the time it takes to get from the 1st floor to the 5th floor, you're gold. You've got it.
Right. But then how do you balance that sort of, you know, you can have a pretty picture that doesn't really convey anything because there's not enough information to conveying too much. So how do you get that sweet spot of 5 seconds?
Well, here's where I find the sentence comes into play as well. So if you can write out the sentence that says what's in it, then you've got content. If you look at the slide and go, I don't really, you know, because again, there's all sorts of Excel, there's fabulous charts. But you're right, some of them just look pretty and don't say much. But if you can write it out in words, I think that's a good check.
Okay, great.
And this is my final slide, just general hints. And again, everybody gets caught up in what they're doing. So it's just good to sit back and remember, spell out your abbreviations. Not everybody works for your company. They may not have the same abbreviations that you do, or the acronyms as well. They may be, you know, your bread and butter, but outside of that, people don't know what they are. Use simple language. Even the smartest people deserve easy-to-read material. And again, I've stated all along, if they're not taking it in right away, they're likely to move on. And this one is specific to the competition, and the judging. You can only evaluate what's written down. We're the same with a reader in a journal article. If you didn't write down your methodology or due diligence up front, you may get dinged for it, or people may question your results. You know, they may question the data quality if you haven't stated that you've done it. I mean, you don't have to say that you run frequency tables, but you can say there was 5% missing data, which we frequently do. And this is how we imputed the missing data. We checked to see that it wasn't systematic. And just cross-reference your numbers to make sure tables add up. It seems like a silly thing, but you know, transposition errors or missing information can throw all your results into question. So below is an example where they've reported an overall repeat rate of 7%, and then after a positive test result, they've got a 10% repeat, and after a negative result, it's got a 25% repeat. Well, if you do the math there, there's no way your overall rate's going to add up. It should be at least more than 10%. So it may have been at the transposition, but I don't know. Should that overall rate have been 17? Should the negative rate have been 2? There's no way of knowing. And just, yeah, have somebody look over what you've written to check for inconsistencies and typos. I know it sounds like a small thing, but it really does throw things into questions for the reader.
Well, those are great hints. Thank you very much. So I encourage you, we've still got a few minutes left, and this is the time where we encourage you to ask even more questions of Jennifer. So she's imparted a lot of information to us all. We've got some good little hints and suggestions on how to improve our analysis, or if you've never done it before, on how to get started. So again, I encourage you to use the Q&A feature. Jennifer, there's one question that I'll probably need to answer. It says, should the Data Impact Challenge submissions be written with the general public as the intended audience, or healthcare providers, or researchers? Maybe I'll take a stab, and then I'll, as a judge, I'll ask you to say what your thoughts are. In some ways, I think it should be written for the general public, because these are going to be published on our site. That said, we know that the judges will, each come with different backgrounds, and so I think you need to take that in mind. As Jennifer was kind of saying, you need to you sort of hone to your audience. So the general public is probably a good rule. Does that make sense, Jennifer?
Yep, that sounds good.
Okay.
There's one other question, if I can answer that one.
Sure.
Okay, so there's a question about how do you know the practice or policy patterns when you're analyzing the data to take it into consideration. I would say the data, you can start with the data and it will give you a hint that something different is going on. Depending on your data, like again, at CIHI we have a largely administrative database. It won't be in the data. We do rely a lot on taking that data, or taking the results and showing them around to people. Again, this is where your stakeholders, this is the relationship. Your stakeholders provide you with information, you provide them with information. Go and talk to people. See if it makes sense from what people see in the field. Sometimes it's a matter of just calling up, or I've Googled facilities. Because something looks wonky and you Google them and you realize they're specializing in renal failure or something like that, but that's not in our administrative data set. So you have to do a bit of investigation, talking to people, researching in some way. It's going to depend on what exactly you're looking at. Like you might find that a certain facility had an intervention. Like they've got really low rates of, like I know North York did a bunch of work on lowering their testing, repeat testing. So if you didn't know that they had done that intervention, their rate would just look really low. But with a little bit of an investigation, you can find out that there was an intervention. So Google.
There we go. While we're still waiting for other questions or final questions, we're going to open up the next poll, or this is the last poll. And so we've asked this question a lot in our webinar series, and we're going to ask it for this one to say, with the Data Impact Challenge 2, how do you see yourself participating? And so we saw that there were a lot of people who are sort of providing data, so we expect maybe this to be a high number B. So the poll is open. Please click your answer and press submit. Jennifer, anything else that you'd like to sort of impart to people aside of final comments as you think about all of the questions that were asked and the information that you presented? What's the key nugget you want people to walk away with?
Oh, that's a tough one. I would say don't get discouraged or don't question your data or overthink your data. I know it sounds sort of contrary, but the reason these questions are out here is because there's not a lot of information. Anything you can provide, even if it's contextual, is still good and useful information.
And I think I'd reiterate that. What we found with the Data Challenge 1, especially when we went through the webinar series to look at those answers, it did raise more questions, but we wouldn't have had those secondary questions raised if we hadn't have done that primary analysis just to get things started. So I think you're right, and that's a good thing for people to remember.
Yeah, this common place for you may not be widely known outside of your area.
Right, right. The poll has ended, so we'll wait for that to come up, and as we do that, I'm going to switch to the final slide and just remind people that the Data Impact Challenge 2 is open until February 29th. Yes, this is a leap year, so you get an extra day in this month to actually submit your entry. It seems like there's a number of people on this call that aren't participating, but it's good that you've learned some further analysis skills. I'd like to thank you very much, Jennifer, for giving this talk, and I'm sure if people have questions outside of this, if they pass them through us, we'll get them to you, and I'm sure if there's some specific questions that you'd be happy to answer them. So thank you everyone for participating today. I'd like to thank the team at InfoWave for making this possible, and we hope you all have a good afternoon.
Thank you very much.
Thanks, Jennifer. Have a good day.
Bye-bye.
Thank you again for joining us today. This concludes today's web conference. You may now disconnect.